In today's lecture, we are going to see on how microorganisms are being classified and the basis that is used for the classification of microorganisms under Introductory Microbiology, Systematics and IKS. Microbes are diversified on almost all the environment like your soil, aquatic environment, terrestrial, under volcanic conditions and etc. So microorganisms were the first living organisms appear roughly 3.5 billion years ago, and it was estimated and calculated that for 3 billion years only microbes were the dominant species or group of organisms that are evolved and existed. So microbes have a very long history, significant, and they are the dominant and highly unpredictable form of your organisms existing in your earth. And the study of those tiny microorganisms are called as microbiology, and microbiology are further subdivided into different branches based on the group of microorganisms they belong to. For example, we have the study of bacteria, that is called as your bacteriology. The study of fungi is mycology, and the study of algae is phycology. The study about your parasites are parasitology, study of viruses are virology, and we have many more branches under your microbiology, which is very fascinating and interesting to study. It was in the year 1665, Anton van Leeuwenhoek was the first scientist who discovered microorganisms, specifically your bacteria and protozoa, microscopically under your microscope, and he named it as animalcules at the earlier times. And when days goes on, he just made further discoveries on those animalcules, stating that they have forward motion and there is no backward movement. They have a head and a tail, and there are internal structures that helps in their movement, and they are diversified in their habitat. During earlier days, the classification of microorganisms was only based on your microscopy, macroscopy and immunological methods. So under your microscopy, we have your staining techniques, that is your gram staining, your capsular staining, and there are many more staining techniques were used to classify microorganisms based on the reactions to which they respond to. And the macroscopic identification or classification of microorganisms was based on your physiological and biochemical characteristics. So under your physiological characteristics, we have the nutritional requirements of microorganisms vary. The carbon sources, the nitrogen sources they use for their survival and growth depends on the habitat in which they live, and so the ecological characteristics also comes under your microscopic characteristics. And under your biochemical characteristics, as we all know, we have different biochemical tests that are used for the differentiation of various group of microorganisms. And few tests, for example, your indole test, MR, VP, citrate, triple sugar ion, urease, catalase tests are some of your biochemical tests that are used to differentiate microorganisms based on the reaction and based on the biochemical pathway and the enzyme pathway they produce, which helps in the degradation of the highly required substrate from the environment by the microorganism. Next, your immunological characteristics is specifically pertained to the clinical part of your microbiology, that is the production or production of your antigen or specific antibodies by the particular microorganism. It can also be differentiated based on your precipitation and agglutination reactions to which the microorganisms respond to. But later, after the discovery of molecular sequencing of microorganisms, specifically the genetic material of the microorganisms are being sequenced by sequencing techniques invented by Sanger, and so the classification has got into its next dimension, which is based not only on your microscopy, macroscopy and immunology, but another criteria of classification was also introduced, that is your molecular based classification of microorganisms, which is now essentially performed in many laboratories and in many research oriented industries to differentiate and classify the microorganisms. And this molecular method is now being used widely to discover new varieties of microorganisms, and this molecular sequencing method helps in sequencing the already existing microorganisms DNA and the newly isolated microorganisms, and this DNA is also sequenced. Finally, the two microorganisms DNA was compared and contrasted to arrive at a conclusion that whether the newly isolated microorganisms is specifically a new species or it is a pre-existing one. Next, as we have already discussed, classification of microorganisms are based on microscopy, that is, the different types of staining methods we use. The physiological characteristics or the biochemical characterization is solely based on the enzymatic pathway that the microorganisms has in the genetic material. The numerical classification is similar, finding differences and similarities between microorganisms, and this numerical classification enables your researchers to find out how similar or how dissimilar the microorganisms are. Next is the genotypic classification. We have molecular based classification, which is being done by using various methods like your PCR, RFLP and so on. We have nutritional supplements, that is your carbon source, nitrogen source, oxygen requirements and so on. So this carbon, nitrogen and oxygen requirements are diversified among microorganisms. Specifically, for example, E. coli can utilize your lactose, glucose and mannitol but not sucrose. But the Klebsiella pneumoniae can utilize all these four sugars, and so E. coli and Klebsiella are differentiated here based on the utilization of sucrose or not. So this also helps in the classification of your microorganisms involved. Next is the ecological characteristics. So ecological characteristics solely pertains on the habitat in which the microorganisms is found, and the optimal pH, temperature, salt concentrations required by the microorganisms for their growth also enables us to classify microorganisms accordingly. We'll see on how microorganisms are classified based on the microscopic methods that are available for the classification of microorganisms. Morphological features that are considered for the classification are size. So size plays a major role in the classification of microorganisms, as microorganisms are classified based on the sizes into various types. That is, we have your rod-shaped microorganism, cocci, cocci in chains, cocci in clusters, cocci in pairs. We have bacilli in pair, bacilli in chains. We have corkscrew-shaped bacteria. We have comma-shaped bacteria. We have spiral-shaped bacteria, and so on. And the shape, as I have mentioned earlier, sizes are of different sizes. They range from micrometers to millimeters, and even viruses exist in nanometers. Next is the colony morphology that is involved. So in case of your colony morphology, different microorganism produces different type of your colonies on your agar plates. For example, some may produce your transparent colonies, some may produce your opaque, smooth, shiny, mucoid colonies and so on. Next is the ultrastructure of your microorganisms. So specifically, microorganisms has specific ultrastructures in them. For example, the components that are present in case of the cells of the microorganism. Next is your spore production. So spore production is another important criteria for the classification of microorganisms, that under harsh environmental conditions like your salinity, higher pH, high temperature and nutrient depleted environments, some microorganisms tend to produce endospores, which helps in extending their survival period for almost 10 years, and at once when favorable condition returns, these endospore gets again converted into the vegetative cells and undergoes all the required metabolic functions, which is essential for the microorganisms to be performed. Next, if the microorganism is a spore former, then these microorganisms can further be classified according to the position of the endospore they have. Next, cilia and flagella are the two locomotory organs that are present in case of different microorganisms. So based on the presence and absence of these two flagella and cilia locomotory organs, microorganisms can be classified under motile and non-motile categories. Next is the color. So the color here indicates the pigment that they produce. So some microorganism produces blue-green colored pigment, example your pseudomonas or cyanobacteria, and some produces your red to pink color pigments like your serratia marcescens and so on. So this pigment production is a criteria that helps in the classification of microorganisms. Next we have is the mechanism of their motility. So if the microorganism is a motile one, then based on the mechanism of their motility, in turn your microorganisms can be classified under various categories. Like some microorganisms can produce your forward movement, some have your backward movement, some have your twitching movement, some have your darting type of your motility. Under Proteus you have your swarming type of your motility. So based on the type of motility, the microorganisms can again be classified. Next is the inclusion body they produce. So inclusion bodies are storage granules that are specific to specific bacteria. Some bacteria can produce your sulfur globules, some can produce your poly beta hydroxybutyrate granules, that is your isobium. So these are the storage inclusions that are produced by a specific group of organisms, but not all the microorganisms. So all the above said morphological features helps in the classification of the microorganisms into various groups. For the microscopic analysis of different types of microorganisms are the bright field microscope, dark field microscope, phase contrast microscope, electron microscopy, and fluorescent microscope. So specifically the electron microscopy is an advanced microscope that is specifically used for the identification and visualization of viruses and not your bacteria or any other group of your microorganisms.